Yes, sure. N2p ratio. So N is the nitrogen, P is the phosphate. The N comes from your ionizable lipid. The P comes from mRNA. So think of this way. To form LNP, first you have to comprehend how is this coming together. You have a negatively charged mRNA that comes with your lipid nanoparticle at a certain ratio that causes an enthalpic precipitation to form a vesicle. Those vesicles fuse together to form an entropically driven lipid nanoparticle. N2p suggests the molar ratio of your lipid to mRNA in this is the molar ratio, and how you bring about is based on how many phosphates you have, what type of delivery you have. Now in most of your COVID vaccines that are currently out there, your N2p is 6. For the siRNA product that's out there, N2p is 3. If you're delivering a self-amplifying mRNA, it would be 9. You have to again focus on the bio performance, and you cannot just cut and paste from the existing product, but usually that's a range it would fall in between.
Terrific. Okay, one more on that. What is the thermal stability challenge with the mRNA or liquid component?
Oh, so lipid again.
I'm
Lipids
throwing
have
random audience questions at you right now, Dr. Bhambhani. Thank you.
no. Lipid is again, you have to think in terms of chemistry. Not every lipid is the same. Lipids have different chemistries to it, but generally mRNA is more susceptible to hydrolysis than your lipid. And why is that? Because mRNAs have a 2' hydroxyl, which is next to a phosphodiester bond, so that's more susceptible to a nucleophilic attack, and that renders it more prone to degradation. So between mRNA and lipid, I'll choose mRNA any day.
Okay. All right. So the question around lipid nanoparticles presenting a particular CMC challenge because they're quote unquote more functional than a simple excipient. Is that relatable to what you've just been discussing, or is that a separate conversation? Is there more you can share there?
Yeah, it is definitely more functional. When I use the word functional, so once again, you have to think in terms of the inherent challenge that comes with mRNA platform. There's intrinsic immunogenicity of mRNA by itself. It's susceptible to enzymatic degradation, and there's almost negligible cellular uptake. So LNPs, what they do is they act like a Trojan horse, and this is the most advanced approach for non-viral delivery of mRNA. The way it does is it protects, so it captures the mRNA and protects it from enzymatic attack. It facilitates cellular uptake and also facilitates your endosomal release mechanism. So the way I would say is LNP, once you inject with mRNAs, it transports it to a desired cell. It facilitates uptake, and it also is responsible for endosomal release. And because of those, it's considered your functional excipient, where it's not just stabilizing, it's involved in delivery, it's involved in endosomal release as well.
Okay.
And so if I can just add in that.
Absolutely. Yes, please do.
So when you look at the challenges in the CMC spaces, is as Akhilesh has just described, right? Your LNP is really designed to prevent degradation of your RNA, right? So being able to deliver the mRNA to the cell, the cell of interest. When we think about the challenge we're all facing, it gets back into stability, and your LNP plays a direct role there. So right now, we've seen with COVID and those vaccines that ultra cold storage and supply chains have been developed in order to enable ultra cold storage of that vaccine to be delivered and sent into vaccination and for everyone to receive their jabs. But that nut still has to be cracked. And there is a lot of work that we're all doing to try and identify how we go from ultra cold storage to the ideal, which is about 4C. So when we think about the innovation and evolution that's going to happen over the course of time, one of the biggest changes we're going to see is improvement in the stability at temperature storages that are more consistent with vaccines that we have on market today. So that's something that we absolutely are continuing to work on, and we're all facing that. No one's cracked that nut yet.
Right. Yep. Excellent. We've received a few questions around critical analytics and CQAs. So I'm gonna try to combine a few of these questions here. We'll start at a high level. What critical analytics and CQAs are important to track through the entire process? Dr. Bhambhani, maybe you can start with that one.
Yeah. So once again, to answer those questions, so what is a CQA? Everything starts with the fundamental definition. What is a CQA? A CQA is nothing but anything that impacts your physical, chemical, biological, or microbiological attributes that can impact your product quality. So when they're talking about it, what analytical or what things to monitor? If you know, be it mRNA design or be it LNP or combination thereof that can impact physical, chemical, microbiological, or biological properties of your product quality, those are CQA. So if you have to overcome those, or when we are talking about analytical, mRNA by itself, you have a 5' cap, you have a UTR, you have a ORF, you have a 3' UTR, and poly A. So if you can characterize the percent capping, the poly A, what I'm talking about it is a mRNA design. How you design the mRNA and the critical element of mRNA that are responsible for immunogenic reaction or product quality attributes, that is one. Same thing in terms of product, the size, the morphology, the encapsulation efficiency. But at the end of this, what you want from a regulatory filing perspective is your content uniformity, your purity, your dose, and your potency. If you can ID potency, purity, content uniformity, all the analytical that goes into it, that's what you want to classify. So for the audience, I would say list out all your quality attributes, identify how it impacts your physical, chemical, biological, or microbiological property based on the criticality. By criticality, I mean how harmful it could be to a patient. Decide, is it critical or not critical?
Perfect. Thank you, Dr. Bhambhani. We've received a few questions about assays. How do you assay for mRNA identity? Since I still have you on the screen. There you go, Akhilesh.
Oh, that's again for me. So how do I assay? You have assaying for mRNA purity. You go with dot blot, ELISA. There are specific assays. You're looking at double stranded RNAs that are used. So those are immunogenic. You have once again, I go back to the fundamental. So if you have a uridine-rich single-stranded RNA, that will cause your TLR7 and TLR8 pathway. The purity is you're looking at double-stranded RNA. So you can use an ELISA assay to look at double-stranded RNA. You can have HPLC, you can have CE assays. Those are some of the commonly used assays that could be look into mRNA purity.